I'm John Brewer.
I'm Mardesich.
And this is color,
Vision,
perception.
So another thing that we investigated was which eye was our dominant eye. This is useful for things like shooting guns or looking through monoculars of any kind. And so the easiest way to check for your dominant eye is by taking your thumb and covering an object. So in this case, we're going to use that meter in the background. And then checking which eye, after closing each eye, the image is covered by. Whichever eye the image is covered in, that's your dominant eye. So in this lab, we're going to investigate our color, vision, and perception of our eyes. And so what these screwdrivers show is me, John Brewer. It's my binocular and monocular fields of view. So here you can see my binocular field of view, which is around 70 degrees. This is a recreation from earlier in lab, so it's not perfectly accurate. And then each alternating pair of screwdrivers shows each of my eyes' monocular field of views.
In the next portion of the lab, we observe where the blind spots in our eyes were. The blind spot is caused by the point where the optic nerve has to pass through the back of the eye. You test for your blind spot by looking at this target, and you put the inside dot at the center of your field of vision, and you move the target in and out, closer and further from your face, and find where the second dot disappears. That's where your blind spot is, and it corresponds to the point where the optic nerve passes through your eye.
monochromator with a grating, and what it does is it separates white light into different colors, and we can use it to determine the cut-on and cut-off points of our vision and what colors we're able to see.
In this portion of the lab, we measured the flicker fusion frequency of our eyes for different colors. We accomplished this by starting at a low blinking frequency on an LED and slowly increasing the frequency until we could no longer see any flicker. That frequency where we can no longer see a flicker is the flicker fusion frequency.
So for the final portion of this lab, we were tasked with finding the contrast sensitivity function of our eye. And the way we did this is we were presented with the stimuli, like this. And if you saw there was one of those intervals in which you could see a sine wave modulation pattern. And so the idea here is you pick the interval in which you saw the sine wave modulation. In this case, it was the second interval. And you continue doing that. And the spatial frequency number here and the contrast number here will continue to go down until you can no longer distinguish one of the intervals from the other. At this point, you've reached your contrast sensitivity limit. And so this means that you can't see. This is essentially a measure of the MTF of your eye. So here are the results of today. So we begin here with our dominant eye test. It turns out that both of us have both dominant right eyes and hands. Alli and Matt both have a blind spot range of around 5 inches, while I have a blind spot range of 6, putting me in last place as of right now. And then we only tried my field of view. So my field of view is my left monocular field of view is 129 degrees. My right monocular field of view is 130 degrees. These could be approximately the same due to measuring error, but possible. And then we have my binocular field of view of 74 degrees, pretty good. With glasses, Alli had a visual acuity of 2015, while I had a visual acuity of 2020. Matt doesn't wear glasses, so not applicable. And then without glasses, Alli has a visual acuity of 2160. Matt has one of 2012, which is better than both of us without glasses. And then I have a visual acuity of 2400. They still let me drive, which is pretty cool, so I'm happy about that. And then for part C, this was our flicker fusion frequency. Looks like the highest average flicker fusion frequency numbers were Alli, with 46, 39, and 49. And then coming in next place was actually me, it looks like. And then Matt comes in last place there.
I was robbed.
And then we have our color vision. The largest color vision range actually came from Matt, which is surprising because he is a man. And normally it's thought that women have better color acuity than men do. So Alli actually came next here, and then I was in last place. And then we have our spatial frequency and contrast in the contrast test. And the best person here, it's hard to say because we had different numbers for both spatial frequency and contrast. It looks to be that Alli at a spatial frequency of 8 could see a contrast of 0.68, so actually looks marginally better than anybody else here. So that would be my guess there. And so I'm going to declare the overall loser me.
This is my guy Jesus.
That was like a 4-second video. This is too much setup for a 4-second video.
Wait, did you like go through different frequencies so you could see?
Yeah, I scrolled through it.
Wait, are you still recording?
Apparently so.
Alli, why?
I probably actually didn't do it the first time.
Alli. Whoa, it's like it's really coming out of the page. It's like it's in 3D.